This is an annotated bibliography example. The title of the paper is Effect of Temperature, pH, Enzyme to Substrate Ratio, Substrate Concentration, and Time on the antioxidative activity of hydrolysates from goat milk casein by alkylase. The citation in APA format is in the middle, and the direct access to the PDF is at the bottom. In Canvas, you have a link to the abstract, and there is a link you click that says Download PDF, and it gives you full access to the site. The paper is open source, and so it is accessed for free. The first thing we want to read through is the abstract. The abstract says the effect of hydrolysis temperature 45, 50, 55, 60, and 65 degrees Celsius, pH 7, 7.5, 8, 8.5, and 9, enzyme to substrate E/S ratio 1, 1.5, 2, 2.5, and 3%, substrate concentration 2, 3, 4, 5, and 6%, and hydrolysis time 30 to 240 minutes on antioxidant peptides hydrosylated from goat's milk casein by alkylase was investigated using single-factor experiment in order to obtain high DPPH, radical scavenging activity, metal chelating activity, and superoxide radical scavenging activity. The optimal conditions were hydrolysis time 150 minutes, temperature 50 degrees Celsius, pH 8, enzyme to substrate ratio 2%, and substrate concentration 4%. The hydrolysis time, hydrolysis temperature, pH, enzyme to substrate ratio, and substrate concentration had a significant influence on the degree of hydrolysis, metal chelating activity, DPPH, and superoxide radical scavenging activity on casein hydrosylate of goat milk by alkylase. The results were beneficial for further provide theoretical basis for production of antioxidant peptides. This article was published in a Chinese journal, and the translation to English obviously is great, but not 100% grammatically correct. So if you notice small errors in grammar, it is simply due to the translation. So when we look at this abstract, we can see first the actual paper studied casein. Casein is a protein. They use the protein and the enzyme alkylase. They look at hydrosylate and how hydrosylate breaks down the macromolecule bonds in protein by hydrolysis. They use alkylase to break apart the peptide bonds in the goat's milk. The study looks at temperature, pH, enzyme to substrate ratio, substrate concentration, and time. The optimal conditions they found are time 150 minutes, temperature 50 degrees Celsius, pH 8, ratio of enzyme to substrate 2%, and substrate concentration 4 grams. For our experiment, we look at lactose sugar and lactase enzyme. We study the reaction rate of the enzyme breaking down the sugar. Our study is based on changes in pH and substrate concentration, and we find that our optimal conditions are 7 pH and substrate concentration of 4 grams. Next, we look at their figures. They have beautiful titles with their figures. They have great axes and labels on their axes. They show a the data graphed with line graphs. You can see they all have keys. You can easily tell what the data is representing because they tell you all the information that is necessary to have. They put the second line, or the figure description, at the top instead of at the bottom. We, however, do it at the bottom following the title, which comes after the figure number. When we look at the figures, we can see each figure has only one independent variable, and all other factors are controlled for. So the first figure says the effect of pH on an antioxidative activity in hydrolysates from goat's milk casein by alkylase. So it's the effect of pH on the peptide activity. And you can see, like our graph with the pH, it comes to a peak and it goes back down, right? The next one is on substrate concentration. And you can see, just like in our graphs, it comes to a peak and plateaus or levels off. In some cases, it does go back down as well. But you can see the trends that are similar in the breakdown of these proteins compared with the breakdown of our lactose sugar. All the other factors are controlled for. So when you look, it says when the selected substrate concentration is 2%, enzyme to substrate ratio is 1%, temperature is 50 degrees Celsius, and under the condition of hydrolysis of 2H, pH varies, right? So 2 hours, pH varies. So they told you all the controls, and the only variable is pH in the first figure. In this second figure, it shows they selected pH was 8, enzyme to substrate ratio was 1%, temperature was 50% for 2 hours, and they varied the substrate concentration. There's only one independent variable, even though the paper looked at both. We used their figures to help identify things in our paper as well that we will write about. So the figure for pH shows there are high points, but then their discussion explains why they are there, what they mean in the context of science. So the pH paper part in the discussion talks about enzyme kinetics and the active site of enzymes and how the pH affects enzyme form and function. The figure for substrate concentration, which those brackets mean concentration, so substrate concentration, everything else is controlled for, and the graphs show how it peaks at 4 grams and is below on either side out of that range. In their discussion, they talk about why substrate activity lessens with regard to the diffusion of molecules, the movement of molecules, and the use of water as a solvent because hydrolysis happens in water. So what would we use this for now? Well, we could use the abstract to compare our reaction rate of the breakdown of lactose milk sugar to their breakdown of milk protein casein. We can use the paper to compare our test of pH and substrate concentration with theirs. We can compare the milk protein and the milk sugar because they are both macromolecules in milk and they are both used for human consumption and digestion. The optimal range for both papers, theirs and our experiment, was ours was pH 7, theirs was pH 8. Substrate concentration, ours was 4 grams, theirs was 4 grams. So those are great pieces of this paper to be able to use in your enzyme paper and a way to incorporate citations into the introduction and discussion. Now we could use their discussion. They talk about enzyme kinetics, the active site of enzymes, and how pH affects enzyme form and function. Perfect for a tie-in on our pH as well. They talk about substrate activity lessening with the regard to molecule diffusion, molecule movement, and water as a solvent? Well, water breaks bonds, hydrolysis. We're breaking apart bonds when we undergo a reaction, and we're looking at how the enzymes affect reaction rate. Both papers are comparing these things. Macromolecules breaking down, being affected by an enzyme. It explains in the results why the results are there, why they got what they got, and what it means in the context of science. So their result discussion and their abstract is huge to support all of the things in our study as well. When you write up your annotation, you can use this information from the abstract and the result discussion to help you remember what you liked in this paper to use and cite in your own paper.